Hello friends and welcome to my channel Sequential Biology. In this video we will discuss about structure of egg in different species. We will discuss the structure of egg in sea urchin and also the frog or the amphibia and the insect egg and chicken egg and human egg. Remember, the egg is nothing but this is a type of cell, and egg is a huge cell. This is a large cell, and probably this is the most largest cell in our body. And since egg is a cell, so there are present protoplasm, and this protoplasm also contains nucleus and the cytoplasm, and this protoplasm is surrounded by plasma membrane. But the important point of the structure of egg is that this plasma membrane are surrounded by many different type of membrane in different species. And whenever you want to study the egg in different species, you should understand what type of membrane are found in different species. Now you can see this is the structure of human egg, and this human egg also contains the nucleus, but this nucleus is not fully formed nucleus. Actually, no nuclear membrane or the nucleolus or the so-called nuclear structure cannot be found in this human egg. This is a ovulated egg, and you know the ovulation occurs about 14th day of our menstrual cycle. But this ovulated egg remains arrested at second meiotic division of metaphase. And in this situation, the nucleus appears as the spindle. No definite nucleus can be found in human egg. And the protoplasm or the cytoplasm is surrounded by a membrane, just like any other type of cell. Since it is a cell, so there must be present the egg membrane or the plasma membrane. But this egg membrane or plasma membrane are usually known as vitelline membrane. Just outside of this vitelline membrane, there are present another membrane which is known as zona pellucida. And in between this vitelline membrane and the zona pellucida, there are present a space and which is known as perivitelline space. And in this perivitelline space, the first polar body are present. First polar body reside in this perivitelline space. Outside of the zona pellucida, the corona radiata are present. And during time of fertilization, the acrosomal enzyme of sperm will break all of this membrane sequentially, and ultimately the membrane of spermatozoa fused with the membrane of egg. So here the outer membrane of human egg is corona radiata, and next zona pellucida, this is the second layer, zona pellucida, and innermost layer is egg membrane, and which is also known as vitelline membrane, and this vitelline membrane covered the protoplasm or cytoplasm. And in unfertilized egg, there are present many granular structure which are known as cortical granules, and these cortical granules reside inside plasma membrane. But when fertilization occurs, these cortical granules will be released into the perivitelline space, and this will form a fertilization membrane which prevent the polyspermy. Then this is basic structure of human egg. Now we see the description about the human egg. The size of egg or the ovum varies greatly in different animals, and the size depends on the amount of yolk present in different species. The different amount of yolk can be present. The egg of the reptiles, birds and the monotremes have a coating and which is known as albumin. And the ovum, then the human egg or the ovum is shed. Ovary is not fully matured, and it is the secondary oocyte. Human ovum is released from the ovary during 14th day of our menstrual cycle, and this ovum is in the form of secondary oocyte form, and later it will be transformed into oocyte and then matured ovum just after the fertilization. Human egg is surrounded by zona pellucida, and it is situated outside of the egg plasma membrane or the vitelline membrane. Then the vitelline membrane and the egg plasma membrane are two names of same membrane, right? And between the cell membrane or the vitelline membrane and the zona pellucida, there are present perivitelline space, and in this perivitelline space, the first polar body lies in this space, and the some cells of the corona radiata can be seen sticking to the outside of the zona pellucida. This is the zona pellucida, and the some cells of the corona radiata sticking with this zona pellucida. And just after the vitelline membrane, there are present cytoplasm or the vitellus, and the no nucleus can be seen, a spindle is present, and the cortical granules are present just below the plasma membrane. So this is the plasma membrane. Just below the plasma membrane, the cortical granules are present in an unfertilized egg. Now we study the structure of chicken egg. You know, there are present many membranes. The chicken egg is yolk laid and a huge amount of yolk are present in chick egg, and so this is megalocecal egg. And this yolk pushes the active cytoplasm towards the periphery, and this active cytoplasm or the blastodisc reside in the top of yolk, right? And this active cytoplasm or the blastodisc and yolk are surrounded by a membrane which is known as plasma membrane. Then this is the plasma membrane. This is innermost layer. This is innermost protoplasm of the chicken egg. Just outside of this chicken egg, there are present a calcareous shell. So this is the calcareous shell. Many porous structure can be found. This is the first layer from outside to inside, calcareous shell, and this protect the egg, right? And just after the calcareous shell, there are present two membrane, the inner and the outer shell membrane. So this is the second membrane. And just inside of this shell membrane, there are present third membrane which is known as egg white or the albumin. This is the albumin. And you know, this is mainly made up of protein. Just inside of this albumin or the egg white, the vitelline membrane is present. So vitelline membrane here is separate from the plasma membrane, but in case of human, the vitelline membrane and the plasma membrane are same structure. But in case of chick, the vitelline membrane is a separate membrane. Inside of this vitelline membrane, there are present the last membrane, and which is known as plasma membrane. So there are present 5 membranes in egg: the calcareous shell or the outer membrane, and then the inner and the outer shell membrane, then egg white or the albumin. This is also another type of egg membrane, and inner side is the vitelline membrane, and the innermost layer is known as plasma membrane. And this plasma membrane covered mainly the yolk and the blastodisc, and this blastodisc is a disc-shaped structure, and this is active cytoplasm. And due to presence of huge amount of yolk, this blastodisc reside in a small region of this egg, actually in the top of yolk. And you know, the chicken egg have the two ends. One is the blunt end. So this is blunt end, and another is the narrow end. So the outer membrane is the calcareous shell membrane. This is a diagram of a chicken egg. Then the calcareous shell, this is the first membrane, and next the outer shell membrane is present, and this is the inner cell membrane. And in between outer and inner shell membrane, there are present a space, and which is known as air space. And this outer and inner shell membrane cover the albumin. So this is the albumin, and this albumin form a chalaza-like structure. This is a chalaza, this is known as chalaza. And inside of this albuminous layer, there are present vitelline membrane, and this vitelline membrane is covered by plasma membrane. And here you can see the yolk, and the two regions of the yolk can be found in chicken egg. One is yellow yolk and another is white yolk. And due to the presence of the yolk, the active cytoplasm just present in a small region, and this active cytoplasm are known as blastodisc, and this reside at the top of yolk. Now we study the description of this chicken egg. So shell, the outermost layer of the chicken egg is a calcareous shell and contain large number of pores and filled by collagen-like protein. And next, the shell membrane next to the egg white, the two layers of the shell membrane are present, and both the shell membrane enclose a large air space towards the blunt end of egg. Then this is the blunt end, and in this blunt end, a large air space are present in between the two shell membrane, right? And this is made up with keratin. And the next layer is albumin. Below the inner shell membrane, there are present egg white or the albumin. And this albumin is also a type of egg membrane. And the albumin is differentiated into thin outer layer and the dense inner layer. And the denser part of the egg white form a spiral strand which is called as chalaza. Then this albumin have the thinner outer layer, and the inner layer is thick. And this thick inner layer form a spiral-like structure, and which is known as chalaza. Then this is chalaza. And 85% of the egg white is water, and the rest 15% is a mixture of several proteins, mostly albumin. Water. The egg white, which is also known as albumin, this contains 85% of water and 15% protein. And egg white provide the nutrition, serve as a water store, and protecting the embryo from the chemical and mechanical injury. And then just after the albuminous layer, there are present vitelline membrane and the plasma membrane, and this vitelline membrane or plasma membrane are separate in chick egg. Below egg white, the vitelline membrane and which is converted to form the zona pellucida present, and the vitelline membrane surrounds the egg plasma membrane, right? And the plasma membrane, the active cytoplasm is called the blastodisc, and the eggs of the hen are polylecithal, macrolecithal and the telolecithal, and most of the space of the animal and the vegetal pole is occupied by yolk, and the yolk and the blastodisc are bounded by plasma membrane. Then any of the egg have two polarity. It has the two pole, one is animal pole and another is vegetal pole. An animal pole is usually yolk free region and the vegetal pole is usually yolk laden region. And since yolk prevents the cleavage, so during the time of cleavage the many cells are formed in this animal pole region, and this will form a huge number of small blastomere. And since in vegetal pole the yolk are present, so cleavage rate is very slow in this region, and this will form many macromeres. The yolk, most of the yolk is liquid and the 23% is solid, and which is known as yolk sphere. And the yolk contains 48.7% water, 32.6% phospholipid and the fats, 16.6% protein and 1% carbohydrate. And the proteins are chemically closely related to the amphibian yolk phosphatidyl and the lipophytolin or the levatin, and the fatty portion about 50%, right? But the fat are neutral fat. Now we see the two types of egg. One is sea urchin egg and another is frog egg. And the sea urchin egg is very important because fertilization is usually studied in this type of echinoderm. And here in this sea urchin egg you can found a cell. So this is a cell, and in this cell the nucleus is present, and the protoplasm is surrounded by plasma membrane, and just outside of the plasma membrane there are present a single layer and which is known as egg jelly. Then in sea urchin egg the two layers or the two membranes are present, just plasma membrane, and outside of this plasma membrane the egg jelly are present, and this egg jelly contains different type of chemicals. And in sea urchin the fertilization is usually external fertilization, so the sperm must be attracted towards the egg of definite species, and this egg jelly plays a vital role for recognition of the sperm to the ovum. Then this is sea urchin egg. Now the frog egg. The frog is also known as amphibia, and there are present two regions, the animal pole and the vegetal pole. And the frog egg contains a jelly-like structure. Just outside of the frog egg there are present a jelly-like structure, and for this reason the frog egg are very much sticky, and it can attach with any substratum due to the presence of a jelly-like albumin. Just inside of this jelly-like albumin the vitelline membrane is present, so vitelline membrane and the plasma membrane are separated in case of frog egg. And inside of this vitelline membrane there are present plasma membrane. This is the plasma membrane, so this is the third membrane. From outside to inside the third membrane is plasma membrane. Then the jelly-like albumin is the first membrane which covered the total egg, and next layer is vitelline membrane, and inside of this vitelline membrane the plasma membrane are present, and this plasma membrane covered the protoplasm. Usually most part of the protoplasm contains yolk, and the vegetal pole and also the few regions of the animal pole also occupied by this yolk, and show the active cytoplasm are present in a very small region of the animal pole, and this active cytoplasm are called as blastodisc. And apart from this there are present many pigmented granules in the animal hemisphere. Then this is structure of frog egg. Then we have learned about structure of sea urchin egg, frog egg, human egg and the chicken egg, and in next video we will discuss the different type of eggs. So thank you, and that's it.